Stem cells are a central element in tissue engineering and regenerative medicine due to their capacity for self-renewal and differentiation into multiple cell lineages. They're defined as immature and undifferentiated cells that can divide asymmetrically, giving rise to one cell identical to the parent cell and another more specialized cell called a progenitor cell. This property guarantees both the maintenance of the cell pool and tissue regeneration. Furthermore, they are characterized by remaining in a reversible quiescent state for much of their cycle, which limits the accumulation of mutations and preserves their biological potential. In this context, cellular potential allows stem cells to be classified as totipotent, pluripotent, multipotent, and unipotent according to their differentiation capacity. Likewise, the behavior of stem cells depends closely on their microenvironment, called the cell niche. In fact, this niche regulates proliferation and differentiation through specific molecular signals and cell contacts, varying according to the tissue, as occurs in bone marrow, intestine, or hair follicle. Regarding cell types, embryonic cells exhibit high plasticity but present ethical and immunological limitations. Induced pluripotent stem cells, iPSCs, offer broad therapeutic possibilities by reprogramming adult cells, and adult stem cells act as tissue-specific reservoirs. Therefore, a thorough understanding of their biology, regulation, and applications is essential for designing safe and effective regenerative strategies in the field of tissue engineering.